All right, so happy Wednesday, November 18th. Just some heads up, hybrid students, you will have another quiz online due November 29th, and it will be over chapter 26 and 27. Everyone else, Friday or Thursday, the week before Thanksgiving, so that means November 19th or November 20th, we will have our quiz in class over neurons, or not neurons, I'm doing neurons in AMP1, over nephrons in chapter 26, and that'll be a 20-point quiz. Thanksgiving week, I will have class on Monday with the Monday noon class, and we'll continue to press on with finishing urinary and potentially starting pH balance, water balance. No class on Tuesday the 25th, and of course Thanksgiving break. So for the week, again, the hybrid students, you have a quiz due by the end of the week, but everybody else, you need to listen to these lectures, review chapter 26 and chapter 27, and we have lab on Thursday the 20th and Friday the 20th, Thursday the 19th is reproduction. So you need to start looking over for lab practical purposes, urinary reproduction, and then getting ready for the rat dissection. Okay, so we're just moving along. All right. So where we left off last time was we finished about glomerular filtration. So at this point, the first step in urine formation is getting a filtrate from the blood plasma. Typically that filtrate is going to be based upon blood pressure versus oncotic pressure between the blood pressure in the glomerulus and the water pressure in the capsule versus the oncotic pressure of the blood and the oncotic pressure of the fluid in the capsule. Filtrate initially is very similar to plasma minus big proteins and blood cells and platelets and things like that, just like what we see with lymph fluid. All right, and filtrate, once it enters the PCT, now we're going to start pulling back and reabsorbing the main components of items and things that we want to keep in water, and then not worrying about and secreting into the filtrate more hydrogen ions, more urea, more potassium, more of the things that we need to get rid of. Okay? So again, filtration, how much we filter normally is going to be based upon the blood pressure of the system. And in a normal blood pressure and normal afferent, efferent constriction, we're looking at filtration rates of about 125 milliliters per minute. Okay? And then we return about 99.9% of that. Okay? We can increase glomerular filtration, recall, by increasing total blood pressure. We can do that through systemic or autonomic means, and that will usually cause a little bit more filtration because more blood supply to the kidneys. And then we can increase glomerular filtration specifically at a specific nephron by manipulating afferent, efferent arterioles, and we can do that through the intrinsic mechanisms, the intrinsic glomerular feedback mechanisms. A little bit of sympathetics will alter, again, afferent, efferent constriction, and then hormones such as angiotensin II, the renin-angiotensin pathway. Okay? All right, now we've got to filtrate. We've got to filtrate. Our goal is to get 99% back, and then of that 0.1% we're losing, make sure it's rich in waste products and unwanted materials. Okay, so this part of what happens to the filtrate as it flows out of the capsule into the proximal loop and distal convoluted tubules is about reabsorbing all the wanted items, secreting, and leaving in the filtrate unwanted items. What do we want to keep? We want to keep usually all of the organic nutrients. So we want to keep the lactate. We want to keep the glucose. We want to keep amino acids. Okay? We just don't want to keep too much. So if we have too much, we might not have enough transporters working at an efficient amount or rate to retain all of it. Or we might have too much of maybe some of the excess proteins or amino acids, and we might go ahead and, again, if you're on a high protein diet, you might find your protein-rich urine. Okay? We want to keep a large portion of the water, and we want to keep a large number of our ions, specifically ions that are hard to get and that play an important role in the intracellular, extracellular environment. Okay? A lot of what is done in the proximal tube is, again, based on those Tmax, so the amount of transporters we have and the rate at which they work. Later on in the nephron, everything is hormonally influenced. Water in the initial parts of the nephron is just based on osmosis, so we look at where is the high concentration of solutes versus low, and water just follows from low to high, solute concentration. But towards the distal parts of the nephron, we're going to, again, hormonally and manipulate water solubility, water movement, and manipulate how much water we keep or lose. At a lot of different places on the nephron, transport is either going to be through active, so requiring ATP. ATP might be required to make endocytosis, so to make maybe vesicles for bringing things back into cells, or it might be to, again, bring something against the brain, against its concentration gradient. Some things will move passively. Just again, like water, will move based on osmosis in the proximal convoluted tubule, so there's no energy required to take the water into the cells and move it into the blood supply. 65% of the reabsorption of tubular filtrate occurs in the proximal convoluted tubule. So you get over half of what you want to keep right away. And then of what's left, whether or not you keep it or not is going to be heavily influenced by hormones and by solute concentration. Okay, and so what your urine looks like is more of getting back 65% initially, and then of that 45 left, whether you work really hard to get the rest of it or work really hard not to get the rest of it. Okay. For the PCT, sodium reabsorption is going to be by active transport. All right, we're going to let sodium go from the filtrate into the cells because, you know, sodium wants to go into the cells because there's a low concentration of sodium inside cells. We're going to learn that in chapter 27. But we want to keep sodium coming into the cells because sodium is co-transporting glucose, co-transporting amino acids, co-transporting, meaning the transporter only will bring sodium and whatever else is tied to the transporter into the cell if both items are bound and sitting in their little docking station. So to keep the movement of amino acids, lactate, glucose happening, sodium needs to stay low concentrations in the cell. So there's a gradient to pull it in. And so sodium is then actively transported, in many cases forced out of the cell at the membrane, basement membrane side of the cell and pushed into the interstitial fluid. And in that movement of sodium out of the cell into the interstitial fluid, in many cases the potassium is pumped in. And so if you remember, it's usually 2 sodiums or 3 sodiums for 2 potassiums. It's an unequal gradient, and it requires ATP to do that. Okay? And again, at the PCT, water is going to be absorbed based upon osmolarity and gradient of solutes, and there's no really fine regulation of water movement. Just water happens, 65% of what is filtered is reabsorbed, and then we spend the rest of our effort and energy on that other 45% of what's left, making sure what's left is going to be rich in urea, rich in potassium, rich in hydrogen ions, rich in excess amino acids, excess creatine and things that we just don't want to keep, and then getting back sodium and chloride and the water that we do want to keep. Okay, so let's focus just first at the PCT, the proximal convoluted tubule. For most of our nephrons, which are cortical nephrons, the PCT resides in the cortex of the kidneys. The cortex of the kidneys is going to have a solute, interstitial solute concentration of about 300 ions and solutes per liter of water. And the plasma of your blood is typically, again, 300 milliosmoles, so that for every liter of water, there's about 300 molecules dissolved in there. Okay? And when we take our filtrate from the plasma, it's also starting at 300 milliosmoles. So we're starting at a situation where the osmolarity is all about the same, and we are going to then actively pump, actively move nutrients from the filtrate into the cells, and then from the cells into the interstitial fluid, and from the interstitial fluid potentially into the blood supply or into the lymphatic system, and retain 65% of what was taken. Okay, so we're going to reabsorb almost all of our organic nutrients. So this is the only place in the nephron where we have transporters for amino acids and transporters for lactic acid, transporters for glucose, transported for organic molecules that could potentially be used to make proteins, RNA, DNA, or energy sources. Okay, we're going to do most of our ions. And yes, we are going to absorb potassium here, and we are going to absorb bicarb, and we are going to absorb sodium and chloride and iron and magnesium and copper and zinc and all those things. All right, and the one hormone that has an effect on the PCT is angiotensin II. And angiotensin II can stimulate these simple cuboidal microvilli-rich cells to just make more transporters, put them on their membranes to bring more sodium into cells, and then pump more sodium out of the cells to, again, facilitate retention of more sodium, more water, and more items. Okay. Again, water is only going to move based upon as things are pulled out of the filtrate, the cells and the interstitial fluid are going to become more solute-rich with sodium and chloride, and water is going to follow that. Okay. There's a lot of passive reabsorption of many of the ions, and there's a little bit of secretion more of hydrogen. So as those sodiums are leaving, some hydrogens are taking its place, and then and that's helping us eventually get rid of more acids that we have made through metabolic and respiratory means from other chemical reactions in other areas of the body. So to put it in a picture, the PCT, the proximal convoluted tubule, is going to have these simple, simple cuboidal mitochondrial-rich, high in microvilli cells. So there's a lot of membrane interacting with the filtrate. Initially, this tubular filtrate is plasma, just minus a lot of the big things. But immediately as the fluid is in the proximal convoluted tubule, items in the filtrate, such as sodium, such as all these organic molecules, such as some of the ions, are going to interact with different types of protein membrane-bound channels. And these channels are going to utilize, in many cases, the gradient that sodium tends to be low in the interstitial or in body fluids, are higher here, so there's usually high amounts of sodium here and very low amounts of sodium inside the cell. And so sodium wants to come into the cell, just like it does in the neuron, just like it does in the muscle, except we don't worry about the membrane potential changing here. What we worry about is we don't want sodium to accumulate. So part of what we do is if we're bringing sodium in with transporting proteins at the side of the cell facing the filtrate, facing the lumen of the nephron, we tie the sodium coming in to bring in other important molecules. And then to keep the concentration of sodium low, we pump. So we use active transport, so ATP is expended to get sodium out of the cell. And that sodium then accumulates, again, back in the interstitial fluid, back in the plasma, all right? And again, to kind of keep somewhat positive negatives compensated, we pump potassium into the cell, where potassium levels are usually high inside a cell, okay? All right, we use energy again to help sodium pump out and secrete hydrogen ions. We use carbonic anhydrase to let us potentially retain more bicarb and get rid of more hydrogen. Therefore, more carbon dioxide is undergoing and binding with water to form hydrogen and bicarb, and more hydrogen is being lost in the filtrate, and more bicarb is becoming part of our plasma, and therefore making the plasma around 7.4 for slightly basic for a pH level. Okay. Again, just another picture of the proximal convoluted tubule. Again, showing the importance of that because there is low sodium inside the cell, we use that gradient of high to low to pull in sodium, to pull in a lot of different molecules. And that lets us reabsorb 65% of what was taken initially from our filtrate. All right? And then we use sodium being pulled in to help secrete more hydrogen. We use that this hydrogen is going to be lost to help us bring in more carbon dioxide to form more carbonic acid, so we have more hydrogen to secrete out. And then we retain the bicarb, which helps us maintain our buffer of 7.4 in our plasma. Okay? And then to keep concentrations of sodium low, we pump. We pump 3 sodium out, 2 potassium in, and so that way we keep low sodium concentrations inside the cell, facilitating then all of the reabsorption that's occurring from the filtrate of lots of different co-transporters and organic molecules. Okay? Now, coming back to reabsorption, remember that if something is reabsorbed, in many cases, it has to have a protein channel. It has to have some type of pore, channel, gateway that can let the item move from the filtrate across the membrane into our simple cuboidal cells, and then from those simple cuboidal cells into the interstitial fluid and into the bloodstream. Okay? So there are a finite amount of proteins on the membrane, and each of these proteins, just kinetics-wise, can only work so fast, right? So they can only potentially move a molecule from outside the cell into the cell at a certain rate. And that rate that they work is known as their Tmax. So what this ends up being for most things is not hugely important, but for glucose, it's extremely important. In your proximal convoluted tubule, the glucose transporter, the transporter that will work when sodium and glucose are bound to the protein, and because sodium and glucose are bound, allows that protein to undergo a conformational change and take a sodium ion and a glucose molecule from the filtrate and move it inside the cell. That protein only works at a certain frequency, a certain rate. And there are only so many of those sodium-glucose transporters located in the proximal convoluted tubule. And so if every one of those transporters is working at its maximal capacity, you can only carry into the cells 300 milligrams per deciliter of glucose equivalent from the plasma. Which means if your glucose levels in your blood are over 300 milligrams per deciliter, when you take a sampling, a filtering of that, there is going to be more glucose in the filtrate than you have transporters, and the rate the transporters work, they could get that glucose back into the bloodstream. So you are going to end up at the end of the proximal convoluted tubule with glucose left in the filtrate. And that might not be a huge deal in a once or twice every now and then scenario, but it's a very big deal if it happens continuously, because that's another solute that we never recover. It's another solute that changes the osmolarity of the filtrate later on when we're trying to manipulate the osmolarity of the filtrate, and can lead to water being retained in the filtrate, water being more robustly retained as urine and being lost by our system. Okay? So how much glucose you keep is somewhat a function on how many of those receptors, those channels, those proteins you have, and what's their maximal capacity. And we know 300 is the magic number for blood glucose. Typically, normal glucose levels are not usually above 150, 170, 200. Okay, if we just ate a meal, they might be over 200 or might be close to 300, but they're usually not that high. So this is usually with diabetics who have uncontrolled blood glucose levels that see continuously high glucose levels, high losses of glucose in the urine, high amounts of then water loss, and that leads to the symptoms of urine output and thirst. Okay? Now, there are some substances that when we take out of the bloodstream, there are no proteins for them. There's no way they can physically get from the filtrate back into the body because there's no transporter for them to be transported back into the body. Right? And creatine is an example of this. So when people take creatine supplements, the creatine that's taken into cells and utilized as creatine phosphate and helping them store a little bit extra ATP is great if they can, you know, get it into their cells. But whatever creatine level gets filtrated from the plasma is not getting into the body because there's physically no way for creatine to come from the filtrate into those simple cuboidal cells, so there's no way for creatine to be reabsorbed. Okay? And then there's a lot of other things that can't be reabsorbed and a lot of toxins and chemicals that we look at. And part of the reason why they're not reabsorbed is they're not lipid soluble, so they can't move across the membrane, and they don't have a protein for them. Okay? All right, so let's review. The proximal convoluted tubule is a big site for reabsorption. There's a little bit of secretion, mainly hydrogen ions. Most of the absorption that is occurring is tied to sodium concentration and sodium being reabsorbed, and 65% of the reabsorption occurs there. Okay, so what's left is now going to be entering the loop. Now, the loop is all about having part of it being water impermeable and part of it being water permeable. So right away, we are going to have areas of the loop where water cannot leave and become part of the interstitial fluid and become part of the plasma, but ions could be moved. So we are going to have a situation where we could potentially change the solute concentration of what's in the filtrate versus what's in the interstitial fluid and what's in cells. And then there will be areas where we can let water move based on osmosis and solute concentration, but we don't let ions move. And this setup is known as countercurrent multiplier. Okay? And it is so important that it happens and it works. Right? So part of what the loop is doing is moving into the medulla. The medulla, especially for those juxtamedullary nephrons, is going to be an area where the interstitial fluid, as we get closer to the minor calyces and closer to the pelvis, is going to be very rich in solutes. Meaning, for a liter of water, so if you have a liter sitting on there, there are going to be areas very deep in the pyramid, towards the tip, towards the minor calyces, that there would be 1,000 little solutes in solution of that liter of water at the very bottom, versus at the very top, you are only going to have in a different liter of water, in a different area, that other interstitial fluid, there might only be 300 molecules in that solution. Okay, so when you have water going and being exposed to areas of 300 molecules per liter versus 1,000 molecules per liter, when water's down in that 1,000 molecules per liter and what's in the filtrate is somewhere around 300 or 400, it's going to want to leave. It's going to want to leave from inside the filtrate and join the interstitial fluid, diluting it down to make it, you know, less solute rich. Okay. So what the countercurrent multiplier does is we set up a situation where as the loop descends into the medulla, okay, the filtrate entering the loop is at 300 milliosmoles. So if we had a liter of filtrate, that means for the sodium, potassium, the chloride, the hydrogen, the urea, in solution, you would have 300 of those solutes, 300 of those molecules, dissolved into a liter of fluid. Okay? Now, as the loop descends, the interstitial fluid outside of the loop is going to start to see the concentration of solutes increase. So as we move down the loop, it's going to go from 300 to 400 solutes per liter, then 500, then 600, then 700, then 800, then 900. And as we're going down the loop, most of the descending loop is the thin portion, which lets water move from an area of low concentration to an area, or low solute concentration to an area of solute concentration. So what we see happening is water, as it's going down the loop, is seeing the interstitial fluid is very rich in solutes and is going to start leaving the filtration and going to then allow the filtrate to become less and less water, and that 300 original milliosmoles or solutes in that liter of water all of a sudden becomes 400, 500, 600, because physically there's less water. The amount of water is decreasing. So to get a liter, you now have to pool more and more and more solutes, more and more volume, and again, the water's leaving. Okay? So we see that the concentration of what's left in the filtrate is becoming more and more concentrated. Now, as we go and ascend, we're going to see most of the ascending part of the loop is thick and is going to be water impermeable. And so whatever water did not leave the filtrate during the descending part of the loop is now stuck. It is not leaving. But as we go through the ascending part of the loop, we now have on these simple cuboidal cells some proteins and some pumps that are going to be able to pull sodium ions, pull chloride ions, pull those molecules from the filtrate into the interstitial fluid. And by pulling them back into the interstitial fluid, we're making the solute concentration in the interstitial fluid back to being 1,200, 600, 700 milliosmoles per liter. But what's happening is the filtrate is losing its solutes. So the filtrate is becoming more dilute, meaning it's going from pretty concentrated to less concentrated. By the time the filtrate gets to the very tip of the loop, you have pulled enough of the sodium, enough of the chloride, enough of the ions that you want, that what's left is mostly urea, hydrogen ions, some potassium, and a little bit of water. And the concentration in a liter of water is somewhere around 100 molecules dissolved in a liter of water, so 100 milliosmoles. Okay? So the descending, you use the interstitial high solute concentration to pull the water. The ascending, whatever water is stuck at the tip of the loop is stuck. It's not going to ever get out again until we get to the DCT. And so what we do is those solutes that we've concentrated down, whatever sodium chloride is still left in there, again, because we only took 65% of what was originally filtered, we start pumping and pulling it back into the interstitial fluid. There becomes less solutes, and the solutes that are in there are predominantly going to be the urea, the hydrogen ions, the potassium, and the water is going to now contain about 100 molecules per liter. Okay? So again, just showing you, all right? More salt is continuously being added to our interstitial fluid by the PCT. So the interstitial fluid is growing rich in ions, in glucose, in amino acids, okay, everything that's being absorbed by the PCT. So our interstitial fluid starts to be in a liter of water. It starts to be very rich in solutes, and it gets richer, and it gets richer, and it gets richer, and it gets richer as you descend in the medulla. That descension of high solutes in the interstitial fluid for the descending part, which is water permeable, allows water to flow from the low concentration of solutes to the high. As water's leaving, what's left becomes more and more concentrated. At this point, when you start to ascend, whatever water is in this little grouping of filtrate is not leaving. It will be up here, still there. But whatever solutes didn't get taken out here, all right, and have just kind of gotten concentrated down, is now going to, for the most part, be pulled out. Many of the solutes that are pulled out are the things we want. So the hydrogens and the urea are going to remain, and we're going to pull out more of the necessary and important ions. And in many cases, we have to pump them out using ATP. Okay? So by the time things leave the ascending part, there's whatever water was left down here is still here, but we've pulled out so many of the solutes that there is, instead of 300 milliosmoles or 300 solutes per liter, we're now down to about 100 solutes per liter. And we move into the distal convoluted tubule, and now we're going to only take back those ions, those solutes, and that rest of that water if hormones direct us to. Okay? And it's just kind of showing you. Because of what the PCT retains, because of what's retained on the ascending limb, and because of the way that water is not allowed to move because it's impermeable in some areas, we create an interstitial fluid in the medulla that progressively gets more salty, more salinity versus what is in the filtrate. And we utilize that to ensure that we can retain some water in the loop. Okay, just another picture here. All right, and part of what happens is the blood supply, the capillaries, also see that change in salinity. And so the water that is taken out in the descending part of the loop is very quickly taken up by the blood supply and then removed. And so that also enhances our ability to keep its very high solute concentration. Right? Now, secretion. Secretion means instead of keeping something or keeping from something in the filtrate going back into the bloodstream, it's something from the bloodstream going into the filtrate. Okay? And we do a lot of secretion in the DCT, but we have been secreting, so far we saw some hydrogen, some urea, some unwanted materials already being put into the filtrate in higher amounts than what was initially taken from the filtrate of the plasma initially at the capsule. The secretion, the big thing is it lets us get rid of our hydrogen waste. It lets us get rid of nitrogenous waste. All right? And so 40 to 60% of urea is discharged in the urine. Remember, urea comes from amino acids. Anytime we break amino acids down and we end up liberating this ammonia, this NH4, well, urea is a way for us to not build up ammonia and become toxic of cells. And then urine lets us get rid of a lot of urea. Another way is when we sweat, we get rid of some urea. And then, you know, it can be reconstituted in other ways, but we get rid of a lot of this. Right? This is also a way to get rid of excess extracellular fluid potassium, because remember, in the extracellular fluid, we want very low concentrations of potassium. Okay? All right, so the distal convoluted tubule is going to be a big place where we see hydrogen ions as well as carbon dioxide converting into bicarb and hydrogen ions, a way to rid our body of hydrogen ions. Okay? Now, how do we keep the hydrogen ions wanting to go this way? Well, we don't let hydrogen ions reconstitute with bicarb, because as soon as we make bicarb, we get bicarb in the bloodstream, so we maintain again that 7.4 buffer. And just like in the red blood cell, a bicarb exiting the simple cuboidal cell here requires a chloride to come in. And then again, to get that hydrogen out, we use chloride to go with it. To get hydrogen secreted, we retain sodium. So we're still using that gradient of low sodium concentrations here, all right, versus higher here, and then using sodium to be a way to drive out urea, to drive out ammonia, to drive out hydrogens, and retain, again, retain water, okay? And we keep some of that excess bicarb by binding it to sodium bicarb, which is a strong, a weak base, which allows us to have, again, a kind of reserve of bicarbonate. Okay? So reabsorption at the DCT. Much of what we reabsorb is hormone-driven. So the hormones we worry about are aldosterone, ANP, as well as BNP from the brain, right? Antidiuretic, ADH. Okay, and then we have parathyroid and, and calcitonin, right? So these three, they are going to affect water. This is about calcium. Aldosterone is also about sodium. Okay, so aldosterone works at your distal convoluted tubule to make more sodium retained. If you retain more sodium, you are going to have to give up more potassium. So it ends up being you secrete potassium, you retain sodium. And then by retaining sodium, you retain some chloride. And then by retaining sodium and chloride, you retain more water. So then you end up with more water, more blood pressure, more sodium. Okay? It can act a little bit on the thick segment of the ascending limb, but we mainly see it at the distal convoluted tubule. ANP and BNP are how we somewhat block sodium retention and therefore water retention at the loop and at the distal convoluted tubule. And by keeping those blockages of sodium retention and water retention, it's a way for us to dump water and diuretic effect and reduce blood volume, and that will reduce blood pressure. It's not as effective as the keepers of blood pressure and blood volume, but it is a way for the heart to signal when it's being overstretched because there's too much volume, too much preload, and potentially damaging. And then it also helps the brain protect itself from too much accumulation of fluid. All right. ADH is from, again, your posterior pituitary, and it's going to work on the collecting duct. So a little bit of the DCT, but mostly the collecting duct. And remember, the collecting duct is going through the pyramid, so it's going to be exposed again to that interstitial gradient up to 1,200 milliosmoles per liter, 900 milliosmoles per liter. So if ADH is present and you put the water channels, the aquaporons, the proteins that allow water to move from the filtrate back into the pyramid interstitial space, you're going to be able to retain more water from the collecting duct, whether over if ADH is not being present and you don't have those proteins on your collecting duct, so you don't get to retain the water. Parathyroid hormone, remember, is all about increasing blood calcium, and it works at the bone to release osteoclasts, work more blood calcium, so more calcium is released to the blood. Okay, as you're taking away the calcium from the matrix, you're making more calcium and phosphate. So what parathyroid does is help us keep the calcium and rid ourselves and secrete the phosphate. If there's less phosphate, then technically you don't have, phosphate to bind to calcium and make crystals and make matrix. Okay? And that's what these pictures are trying to show you. So in our DCT, all right, when you have aldosterone, all right, we ramp up that sodium is retained, and to keep the sodium and to run this pump to get 3 sodiums, we have to pump in 2 potassiums, and then potassiums end up being secreted into the filtrate. Okay, and typically for the sodium coming in over here is usually tied to bringing in a chloride. Right? Again, just to redefine, and a lot of how the kidney works and a lot of how the water is reabsorbed in the loop, in the DCT and the collecting duct comes back to this chemistry concept of osmolarity. All right, so osmolarity is about the number of solutes, the number of particles dissolved in a liter of water. So right now I have a Slim Fast, like milk, in front of me, and then I have a Diet Dr. Pepper, and then I have water. Each one of these items has a different osmolarity, meaning each one of them has a different amount of solutes dissolved in a liter of that fluid. So if I have a liter of Slim Fast, the osmolarity, because there are proteins, there are fats, there are ions, there are vitamins and minerals, it might have an osmolarity maybe at 2,000 milliosmoles per liter, meaning there's 2,000 potentially solutes dissolved in a liter of this water of Slim Fast. Okay? Dr. Pepper might not be as rich as Slim Fast, but it still does have some sodium. It still does have potentially some caramel color, some aspartame, some phosphoric acid. So while it might not be as rich in osmolarity, it might still be 200-300 milliosmoles per liter. So a liter of Dr. Pepper would have about 300 particles dissolved in that liter of water. And then pure water, all right, if it's not distilled, deionized, okay, and that's what you'll hear these terms when you work in a lab, where you really filter water to remove all ions, all electrical properties or anything that could be in it. Water might have, 20 random ions, like maybe a random sodium, a chloride, a calcium, dissolved within a liter of it. It might not be pure water. Okay, that's one of the things when they say it's filtered, it should be pure water. So there should be potentially 0 solutes, 0 particles, 0 extraneous molecules other than just pure H2O in that liter. Okay, and so what happens is if I was to set up a situation where the Dr. Pepper and the Slim Fast could interact and water could move between them, because the Slim Fast is so rich in solutes, I would expect that some of the water from the Dr. Pepper to move into the Slim Fast, and the Slim Fast would become more diluted, meaning it might go from 2,000 milliosmoles to 1,500 milliosmoles as the water moves in. And that's what osmosis is, okay? So our body water, so our extracellular fluid, our lymphatic fluid, our plasma, normally wants to have about 300 particles for every liter of water. All right? So your body works, your system works to try to maintain about 300 particles dissolved in every liter of fluid, of body water that you have. And so what the kidneys try to do is maintain this ability, so as the fluid comes to the kidneys, if we need to take more solutes out, we do. And we make concentrated urine. And by taking all these excess solutes out, we maintain 300 milliosmoles throughout the system. If we start to take in maybe too many fluids and our milliosmolarity starts to become diluted in our system, we try to then remove more water so that way we can maintain the right milliosmolarity in the different organs, so we don't have too much fluid like lymphedema or too much fluid pressing on connective tissue and causing problems, causing compartment syndrome. Okay? And so the kidneys work to keep 300 milliosmoles. Now within the kidneys, because there are places we want to retain or not retain water, this is where we see in the medulla and in the filtrate, we set up different areas to have higher or less than 300 milliosmoles. And by doing that, we can facilitate more water retention or less water retention. Okay? So again, when you have a dilute urine, that usually means that the nephron took some fluid and you want to rid yourself of more water than necessarily solutes. So you want your urine to be less than 300 milliosmoles. So if we take a liter of your urine, we would expect to find less than 300 particles dissolved in that liter of water. To do that, what you're going to do is you're going to let the PCT do its thing. The PCT is going to get 65% of the filtrate. Of what's left, you're going to let it move through the loop. And through the loop, you're going to let yourself retain a lot of the water in the descending limb and a lot of the sodium, the chloride, and the potassium in the ascending limb. In the DCT and in the collecting duct, you are basically going to do nothing concerning the water. So you're not going to try to keep more sodium, and you are not going to try to put extra channels and pathways for water to get back into the interstitial fluid. And so the fluid that is entering the DCT, the water that's coming into the DCT, and the solutes that are in the DCT are going to potentially be that 100 milliosmol level. So it's urea, some hydrogen, some potassium, maybe a few more sodium and chloride molecules and whatever. And that's it. You're just going to let it be lost. Okay? And you might even add, maybe take out a few sodium, but add a few more hydrogen. So whatever you reabsorb out, you quickly secrete the equal amount of particles in. So again, you don't necessarily change the number of solutes in that liter of water, and it stays at 100 milliosmoles. Okay? And this happens when ADH is not present. Okay? So as you can see here, again, we have each little dot probably recommends so many solutes, and you have about 300 solutes in a liter of water, and that's what it would look like, you know, space-wise. And at the end of the PCT, you are now at 300 solutes still per liter of water. As you go into the medulla, remember, the concentration of solutes is high here, and it's getting higher and higher. And so water is being pulled out. But the solutes that were here are not. So they are becoming more and more concentrated. And as we go and ascend, we start pumping out those solutes, but we don't let the water leave. So the water here is lost. It's the same water here. But the solutes have been pulled back in to maintain this high interstitial fluid. And without ADH, there's no more water retention. There's no more ability for water to leave because the rest of this pathway through the DCT and the collecting duct is water impermeable, and that water is lost, and our urine will be less than 300 milliosmoles. Okay? Now, When we want to retain water, because maybe our body's becoming less water and the milliosmoles are beginning to creep up in our plasma, creep up in our brain, creep up in our different body compartments, we're going to want to retain more water to make sure we can dilute it back down to about 300 milliosmoles. So again, we're going to want to retain more of the water at the end of the loop in the DCT and the collecting duct, and we're going to want to make sure that we maybe get rid of more of the solutes and specifically the waste products that we don't want to keep. And this is where ADH is going to play a big role because, again, the PCT does its normal thing. It's 300. The loop is going to work its normal way, pulling hydrogen in, pulling water in, and then keeping whatever water's here, pulling the sodium chloride, maintaining countercurrent. Nothing changes. Once we get into DCT, because ADH and because of the mechanisms in place, we're going to allow, with special pores, with special proteins, water to now be permeable and be able to leave the filtrate in the DCT and the collecting duct. And because the collecting duct passes again through the pyramid where that concentration of solutes per liter is so high, it's going to help us pull in even more water. And so at the end, we are going to end up with even more water back in our blood supply, back in our bodies, and our urine concentration will be extremely concentrated. So this is the summary of the nephron. The nephron we filtered. That was step one for making urine. Then we, throughout the PCT, the loop, the DCT, and the collecting duct, we absorbed based upon the regular number of Tmax values of proteins and how they work, and hormone influences, and absorbed based on osmolarity gradients for water. And we secreted waste and things that we need to get rid of, and those wastes somewhat keep osmolarity at places. So in the PCT we stay at 300, but in other places it allows us to manipulate osmolarity gradients between the interstitial fluid and the filtrate to enhance water retention or water reabsorption. Okay. Now, when we talk about kidneys, one of the big set of drugs that work on kidneys are known as diuretics. And a diuretic is any type of molecule that on the kidney would prevent water being reabsorbed and would increase urine output. So caffeine is a diuretic, because part of what caffeine does is it increases a little bit of our blood pressure and our heart rate, and that makes more filtration and potentially causes more fluid to be lost at the kidneys. Okay? Now, diuretics can work in a variety of ways. Diuretics can be substances that are like drugs, put into our drug system, end up being filtered, can't be reabsorbed like creatine, can't be reabsorbed because they're not lipid soluble, they don't have proteins, become a fixed solute, and therefore change osmolarity a little bit and cause more fluid to be lost because water, not as much water can be reabsorbed with the osmosis gradient. Okay? With glucose's situation. When glucose is too high and it's not reabsorbed all the way because it's too high and there's glucose that ends up in the filtrate later on, it becomes another fixed solute that changes the gradient. So instead of being 300 to 1200, you might be 400 to 1200 or 500 to 1200, and you just don't get as much water reabsorbed. Okay? A diuretic can be anything that blocks sodium reabsorption at any point of the nephron. So if it blocks sodium reabsorption in the proximal convoluted tubule, it's going to block potentially a lot of things being reabsorbed, but it's going to block some of the water retention there. If it blocks sodium reabsorption in the ascending thick portion of the loop, it will block some of the sodium being recovered, some of the changes to the solutes, and potentially the water reabsorption. Again, same thing with the sodium levels of aldosterone. You block aldosterone, you're basically making that drug as a diuretic because you're not then retaining sodium and not retaining water. Okay? And a diuretic can be anything that blocks ADH. This is why alcohol is a diuretic. Alcohol, OH negative, when it gets into your bloodstream, when it gets into your system at the stomach, travels to the brain, blocks the hypothalamus from secreting ADH, and now there's no ADH to put channels on the collecting duct and the DCT, no water retained, urine output increases. Okay? So osmotic diuretics are a specific type of diuretic, and that we see this with diabetes. This is where, because there is a solute that shouldn't be in the filtrate and is there because you had too much of it filtered and it overdid its Tmax, can start to then cause water to retain in the filtrate, retain in the fluid, and be lost. Okay? And so these are some of your different types of osmotic diuretics. Right. Now, we've gotten past the collecting duct. The collecting duct's gone into the minor calyces, the major calyces, the renal pelvis, the renal peristosis is bringing that fluid through the ureter, and that ureter is going to collect that fluid in the bladder. And at this point, we have urine. So once you get past the collecting duct, you have urine. You're not going to get any of that back. All right, and at any given second, every nephron is making a little bit of a different urine and collecting ducts, bringing together a bunch of different, minute little differences between the concentration and the composition and the makeup. But what urine typically is going to be mostly water, and anywhere from 1,000 solutes per liter to 1,200 solutes per liter. But even those solutes are mostly hydrogen, potassium, urea, ammonia. It's not big molecules. So the urine should be fairly clear, and again, because of a little bit of the urochrome and the bilirubin and things like that, slightly pale or yellow. The more concentrated, the less water, so the more closely connected the urochrome, it turns a darker yellow. The more dilute, the more water, the more break between the urochrome, so they don't get to, be as heavily concentrated, they will be lighter. Okay? And again, where's urochrome? Because it comes, remember, some of the red blood cells that spontaneously destroyed themselves and caused some of the hemoglobin and some of the proteins of hemoglobin to be released and broken down in the blood supply and at the kidney are put into the filtrate. Okay? All right. Again, sometimes when you eat asparagus or take certain drugs, urine will take on like a greenish color, or there's even a drug, it made my kids have reddish urine. And again, it just might be because of the way the drug has a color or a protein or something that looks a certain color, and as it gets more concentrated, it makes the urine a certain color. It might even make a smell if that item, like with asparagus, sometimes your urine has a funky smell. Cloudy urine may indicate the presence of bigger things that shouldn't be in there, like big proteins or cells. And it may indicate an infection's occurring and that bacteria are making excess, like waste products that are ending up in the bladder and then in the urethra and then in the urine. Urine, because the hydrogens are being actively secreted, so initially filtrates 7.4 like plasma, but as it goes through the PCT, the loop, the DCT, more hydrogens, more urea, more acids are added. And so pH should be slightly acidic. It can get as low as 4, 4.5, and most people with an American acidic diet, it sits between 5 and 6. Some people will have an alkaline urine. Those people either are very, very well indoctrinated vegan vegetarians who eat a lot of basic foods, and so therefore they're trying to get rid of the load of basic and excess bases that they've taken in, or some of these people might be prone to metabolic alkalosis or respiratory alkalosis, and so the excess bases are being gotten rid of through their urinary system. Most of the time, we're acidic. Okay? But again, vegan diets, a lot of your foods, a lot of your vegetables are very basic foods. Not basic meaning simple, basic meaning have a high content of buffers and bases in them, and so they can actually make your diet more alkalinic, and more alkalinic diet can lead to urine output that actually becomes more neutral or even slightly basic. Most of your urine is actually water and very small amount of solutes. And the key here is, again, from proteins and nitrogen content, we're getting rid of a lot of those nitrogenous wastes that were from the breakdown of proteins and the breakdown of nucleic acids, purines and pyrimidines. Okay? There will be a little bit of ions in there and some other proteins, but blood, red blood cells, white blood cells, some albumin, some of the big proteins, glucose should not be in our urine. And if they are, that indicates a pathology. It might be a pathology at the urethra, because again, you might have a little bleed in your urethra, and that could be the source of the blood. It might be a pathology in the bladder. It might be a pathology in the ureter, or it might be a pathology of the kidney. It doesn't necessarily have to be all the way back at the nephron. There's a lot of space between the nephron and the urethral opening. But those things should not be typically, you shouldn't be bleeding with your urine and big proteins. Okay? And again, what are these ammonia, urea? Many of these things come from amino acids, nucleic acids. Okay? And creatine, again, we just don't have transporters for it. So any excess creatine or any creatine filtered is lost. Okay? Now, the bladder is the site for urine storage. To get urine there, the kidneys transport it down the ureters to the bladder. The ureters are going to be a tube that is going to, like the esophagus, utilize gravity, but when we're laying down or not, upright, and gravity is not available, it's going to have to use peristalsis and other things. Okay? Like other tubes, it has layers. The innermost layer is going to be a mucosa. Some of the ureter is going to be transitional epithelium. Okay? We're not worried about, in the ureter, we're not worried about reabsorption or secretion. We just want a nice barrier where the filtrate stays in the tube and not getting back into our bodies. There's going to be a muscularis layer. Again, muscle is going to be important with peristalsis, with generating some waves and rhythmic contractions to get the fluid to move to the bladder. Okay? And then we have an adventitia for a large part because the kidneys are retroperitoneal. The ureters are going to be retroperitoneal. They are not going to be part of the visceral peritoneum at all, parietal peritoneum, any of that. So it's an adventitia, so a tough connective tissue. Okay? Now, it's a very small tube and a very small lumen. So this is why kidney stones, when you have excessive ions and ions form crystals and crystals rip through the ureters, they can tear and rip and damage and even occlude the ureters, and that can be very painful. Okay. The bladder is located on the pelvic cavity, so it's inside the actual pelvic bones. It's posterior to the pubic symphysis, and it has three layers. It has a mucosa, innermost layer, that's transitional epithelium. It has muscle layers, right? And we're going to specifically talk about the detrusor muscle because the detrusor muscle is going to be what helps to contract and press urine out. And then part of the bladder sits at the base of the peritoneum. So the very superior part of the bladder is actually going to be the parietal peritoneum. But the sides and the inferior part of the bladder are outside the peritoneum, so they are going to be an adventitia. And they are going to be connective tissue that's holding, for females, the bladder in front of the vagina in place, and then in males, it's going to hold it in its spot next to the rest of the rectum and, holding it so it's connected to the penis and all the glands. Okay? In the bladder, there is this triangle, three openings. There's two for in, one for out. So it opens, and they call it the trigon. All right? And it makes the place where the two ureters come in, and then a central place where the urethra goes at. And because it looks like a triangle, it's part of the reason why it gets the name the trigon. Okay? And then your rugae. So when we look at the bladder, because of the way the bladder can expand, it's going to, when it's not expanded, not stretched, the mucosa and the muscle layer are going to make these big ridges and folds like the stomach. And so we are going to see those folds called rugae. Okay? The bladder should never hold more than 1 liter of fluid. Okay, so most of us, our bladders are around maximal stretching capacity, full to the max at 800 milliliters. So a little bit less than 1 liter. All right, our bladder likes to, not be overly full, so it is considered like a safety net for itself that when it's about 500 milliliters, that's where it considers itself full. and it will start trying to empty anywhere between 250 to 300 milliliters. So when it's technically about a quarter full of maximal capacity, maximal stretch, it's going to start thinking, okay, let's get rid of this. Okay? And this is why in car rides you kind of like can hold it and forget it and then add a little bit more fluid to the system, regenerate those stretch receptors, and get the sensation maybe 20, 30 minutes later of, oh yeah, I really do need to pee. And it's becoming more urgent as my bladder continues to fill. All right, so this is a female bladder, okay? We have 2 ureters. This is going to be the most anterior of the items, and the vagina is going to sit behind it. The top is going to be the visceral peritoneum. The sides and the underneath are going to be for the outer layer and adventitia. The ureter or urethra is going to be very small. The trigon is made where the 2 ureters come in and the urethral exit is opening. The muscle layer is going to be the detrusor muscle and some smooth muscle mixed in. Okay. All right. The transitional epithelium is going to be here with the help of the detrusor and the transitional epithelium. Unfilled bladders are going to be collapsed and are going to have rugae. Filled stretch bladders the rugae is going to be less pronounced. For the urethra, there are 2 parts. There's the part that opens to the outside environment, and there's the part where it collects fluids from the bladder. The internal urethral sphincter is the opening between the urethra and the bladder. This is a smooth muscle, involuntary controlled muscle. So this muscle relaxes as pressure builds. So it's a reflex of increased pressure, sphincter relaxes. The external sphincter is going to be skeletal muscle. It is going to relax partly under voluntary control. Okay, so this is what you learn to control in potty training. All right, catheters bypass this and shoot into the urethra and then even into a little bit into that internal urethral sphincter into the bladder. And so what catheters are is they just, as the fluid accumulates, it just continues to flow into the tube and accumulates in the bag. Okay, catheters though are tubing and foreign objects and a passageway for things to get into. So that's part of the reason why we don't like people to be on catheters for long periods of time, because it increases risk of introduction of pathogens. Again, the female urethra can be anywhere from 3 to 4 centimeters long. It's not very big. It is very close for the bladder to interact with the outside environment, so more likely to get UTIs, urinary tract infections here. External urethral orifice is between the vagina orifice and the clitoris. The internal urethral sphincter is between the urethra and the bladder. The detrusor muscle is going to be the thickened, smooth muscle around the bladder that has some involuntary control to contract the bladder to help push fluid onto the external urethral sphincter, which is under voluntary control. All right, so the male bladder is a little different, okay? It's going to be, you know, a little bit tied to a different urethra, and there's no vagina between it and the rectum, okay? And the male bladder, same thing, superior visceral peritoneum, still the detrusor muscle, stereotrigon, still an internal urethral sphincter, internal os, and an external sphincter, but the external sphincter is extremely going to be important of getting fluid into the penis. Okay? So there's 3 regions to know for the urethra, and the urethra for the male gets its name by what area or gland does it pass through. Initially, the urethra from underneath the internal urethral sphincter until the external diaphragmatic urethra is going to pass through the prostate gland. So this part of the urethra that passes through the prostate is known as the prostatic urethra. Okay? The next part is going to be the membranous urethra, which is going to pass through some of the tissues and the membranes of the pelvic floor, and that is going to be right here. It's the smallest one. The spongy urethra is going to be surrounded by the erectile tissue, the spongy corpus cavernosum and the spongiosum, and that tissue, which is capable of being engorged with blood and putting pressure and erecting the penis, is going to be the spongy urethra. Okay. So let's talk about nutrition, voiding urine. Around 200, 250, even for some of us 300 milliliters of urine in our bladder starts to be enough weight, enough fluid to stretch the rugae, to stretch the bladder, and there are smooth muscle and stretch receptors located in the transitional epithelium and in the muscularis layer. And these stretch receptors are going to carry information through the afferent peripheral neurons, the sensory neurons, to the sacral part of your spinal cord. Right? And some of those signals are going to continue up to the brain to be into conscious awareness, like, hey, my bladder's getting full. Okay? And that's going to then let me think, like, oh yeah, I need to maybe make arrangements to go to the potty. All right? And as the signal ascends, it's going to inhibit some of our sympathetic neurons that are trying to maintain sphincters that are closed. Now, once your brain gets the information, okay, I'm going to need to pee, there's going to be further sympathetic neuron inactivation and inhibition. So we can get neurons, smooth muscle cells, under sympathetic control to relax. So some of those internal smooth muscle sphincters will start to relax. And we'll start to stimulate parasympathetic neurons that can get more nitric oxide release to, again, get smooth muscle cells to relax. The result will be that the internal sphincters are relaxed, and then our detrusor muscles and our smooth muscles of the bladder will start to contract. And that'll push the urine from the bladder into the urethra. Now, the external urethra is still contracted. Right? And we've gotten conscious awareness that, okay, my bladder's full, and we start to feel the stretch and the pressure on the external sphincter. And that'll then trigger a different descending pathway and a different effect of getting a motor neuron from our spinal cord to release acetylcholine onto these skeletal muscles, to get calcium release, to get muscle, contraction to be inhibited, and that will then allow us to relax and void urine. Okay. So again, stretch receptors embedded in the submucosa, kind of right behind the transitional epithelium, and in the detrusor muscles are going to sense the bladder getting full. They are going to carry information to the sacrum. They are going to, some of that information, head to the pons, head to the cortex, so you become aware that your bladder's getting full. Another part of what is going to happen is going to be a little reflex to get parasympathetic ganglion in the bladder wall to contract the detrusor muscle, to contract and expel urine past the now, hopefully, relaxed internal sphincter into the urethra. Okay? Now, as the body realizes, okay, I'm full bladder and I'm getting a little pressure on my external sphincter, I'm going to use somatic motor neurons that come down in the sacrum, come down and cause relaxation of the skeletal muscle here to let that urine expel. And again, we want to get more contraction of the detrusor muscle to get more of that fluid pushed out of the bladder. So that is the urinary system, you guys. And I will do pH and water balance on Friday. Also note that this week's lab is all about reproduction. So in lab, do expect about 20 minutes where we're going to start reviewing again the bladder, the urethras between males and females, but we're going to also start hitting on uterus, ovaries, testes, and spermacords and all that fun stuff. So see you in class. If you have questions, of course, email me and ask them in class.